Hello. Hi everybody out there in Zoom land. My name is Peter Dearden from the University of Otago and from Genomics Aotearoa, who are delighted to host this Asia Pacific seminar. I'm just here for your entertainment at the start of the thing. The moderator for this seminar is Mark Tanaka, who is a mathematical and computational biologist at UNSW. So I'll pass on to Mark, and he will introduce our speaker.
Hello, everybody. Welcome to the Asia Pacific Genetics Seminar Series. Our speaker today is Associate Professor Kim Handley. It's a great pleasure to introduce her. Kim is a microbial ecologist in the School of Biological Sciences at the University of Auckland. She leads an environmental metagenomics project on aquatic microbiomes with Genomics Aotearoa and serves on the science leadership team. She completed her PhD at the University of Manchester and postdoctoral research at the University of California, Berkeley, and the University of Chicago. Her laboratory focuses on genetic adaptations, e.g. salt and temperature tolerance, and metabolic processes, e.g. nitrogen cycling, in microbial communities. And this research aims to understand how bacteria, archaea, and associated phage shape and are shaped by the physicochemical conditions of aquatic environments such as streams, estuaries, aquifers, using genomic techniques. So it's a great pleasure to have Kim speak today, and she's going to tell us all about her topic on physicochemical conditions. So I'll hand over to you.
Okay, thank you very much, Mark, and thanks, Peter, for inviting me. So I'm just going to share my screen. Cool, and hopefully you can see the correct screen. Thanks for joining today to listen to this talk, which is a little bit microbial. So I'm going to talk about some work my lab has done in salty environments, so in estuaries and in hot spring environments as well, and how we use environmental genomic data to look at genetic adaptations in those environments. And so why are we interested in salt and temperature? Salt in particular is a major global driver of microbial community composition. So it's often touted as number one, or it's right up there with host association. So whether or not a microbe, a community is associated with, say, a plant or an animal, salt is right up there. So it controls microbial compositions. Other drivers, which are maybe a little bit less strong, are water versus sediment, pH and temperature, although when you consider hot spring environments, pH and temperature really come into their own because of those really big differences that you get in those environments. And so I'm going to focus on salt and temperature. Well, both of these are kind of related in the respect that they require physiological adaptations for protein stability that are related to non-synonymous codon bias. So in other words, changes in amino acids. So they are related. And so I'm going to, before I go on to that, I'm just going to touch on something that is also a bit analogous. It's synonymous codon usage bias. So this isn't the main focus, this isn't the actual focus of what I'll be talking about, but it's also used to identify, or it is associated with different traits in microbial communities. And so synonymous codon usage bias is the bias in codons where you have that bias is where a gene is really highly expressed and the codons are different to what is typically in the genome. And so organisms with that bias tend to have higher growth rates and also habitat flexibility as well. So organisms that exhibit habitat flexibility include pathogens, facultative anaerobes, and also microorganisms that have broad growth temperature ranges. So you see that there. So getting back to salt and temperature. As mentioned, these are the adaptations that are required for salt and temperature involve amino acid substitutions. So in particular with temperature, the enrichment of proline. So proline is an amino acid that confers rigidity to polypeptide chains. So in proline, you have the side chain folded back and bonded with nitrogen to form that lovely ring structure. Over here, hopefully you can see my mouse. And this creates a rigid structure in proteins. So helps to stop proteins basically unfolding in at high temperatures. Same with salt. There's an enrichment instead of acidic amino acids. So glutamate and aspartate are the two acidic amino acids, and can be a depletion of basic amino acids as well, in particular lysine, less so arginine and histidine. So in this case, this enrichment of acidic amino acids helps to increase protein surface solubility and impart a hydration shell to proteins. Okay, so these are not the only adaptations that organisms living at high salt or high temperature have, but they are substantial. So it's more than just the acquisition of a single gene or operon. And this is thought to limit transitions of taxa from one habitat to another. So from a freshwater habitat, for example, to a marine habitat where you have to acquire all these adaptations. So these sorts of switches where an organism might change the type of environment it likes to live in are not thought to happen very often. Okay, so I'm going to start with salt before moving on to temperature. And so there are two types of strategies. One of them is more closely related to this enrichment of, or is typically thought of in association with the enrichment of acidic amino acids, and that's the salt-in approach. So the salt-in approach over here is mostly found in extremely halophilic archaea, also some halophilic bacteria. It's kind of associated more with higher salt concentrations, and it's the most efficient process. So basically, microbial cells just decide to let salt in. They prefer potassium, so they preferentially enrich potassium in their cells, so they have very salty cytoplasms, and for that reason they need their proteomes inside their cell to be enriched in these acidic amino acids. Most halophilic bacteria and moderate or slight halophiles use the alternative approach, which is the salt out approach, and they're pumping salt out of the cell and then stuffing their cell with organic solutes instead to maintain the pressure in their cells. And this is what we're going to be focusing on. Okay, so there is some evidence for these salt-out bacteria with to have slightly acidic proteomes. These bacteria include moderate and slight halophiles. So a slight halophile would include marine bacteria, but it is not yet demonstrated, or had not been demonstrated for brackish bacteria from estuaries. And in terms of using genomics to predict these traits, it's not well demonstrated for marine bacteria either. And so over here we have this this figure from quite a nice study published back in 2010 that used metagenomic data to show this lovely trend, sort of an opposing trend between lysine encoding and aspartic acid encoding at decreasing salinities. So in this case, they focused on really high salinities, and you can see this lovely opposing trend. It's actually linear. But when you get down to marine salinities and very slightly salty salinities, you don't see that trend anymore, and we think that's just because of the sampling resolution there. And so in the study we undertook, we sampled across an estuary from stream, the brackish environment, to the oceans. We had quite a lot of different salinities across this lower part of the salinity regime. This work was largely led by a postdoc of mine, or initiated by a postdoc of mine, Carmen Acudillo Garcia, who is shown here. And what I'll talk about today is a demonstration of the sought-out approach in these estuarine communities, and the enrichment of acidic amino acid encoding for the secretome of these organisms. I'll talk about that, how we found that across compositionally distinct communities and diverse phyla, and also salt adaptations in what we would consider a typically freshwater lineage that has jumped that boundary between freshwater and salt. Cool. So in our study design, we use two compositionally distinct communities, so water and sediment communities. You can see that compositional distinctness here in these taxa plots. So these are colored by phyla. This top plot shows communities at different salinities, increasing salinity across the x-axis in water and in sediment. You can see they're quite different. And this plot over here shows the average phylogenetic distance of these communities relative to E. coli. So you can see again, they're very different. And so we looked at these trends in these two different communities. And what we found when we looked at amino acid encoding was that out of all the amino acids when we looked in water and sediment communities, it was only the acidic amino acids, aspartate and glutamate, that gave us a consistent, significant trend with salinity for both of these environments. So with both of these acidic amino acids, we found that they were lower. The encoding was lower in freshwater. So that's freshwater here in sediment, and freshwater here in water, and higher at higher salinities. So we're going to focus on our charged amino acids now. And so these plots show over here our acidic amino acids, and over here our basic amino acids. And so you can see this trend is really strong again with across salinity on the x-axis, with our glutamate encoding in water and in sediment shown in orange, and with aspartate as well. Less so with these basic amino acids. And this is considering all protected proteins. So that's maybe not too surprising, given we're not exactly expecting the entire proteome of organisms using this salt-out strategy to be acidic. Before I move on to look at the secretome, I'll just point out this linear trend. So as I mentioned in the previous study, they found a linear trend at very high salinities. Here we're demonstrating that a continuation of that trend in low salinity. So on average, each of these points is an average of genomes in a community. When the community composition at a particular environment, as you move up in salinity, you get an increase in the acidic amino acid encoding. Okay, so because these organisms we think are using the salt-out approach, so actually pumping salt out of their cells, we expected the trend to be strongest in the secreted fraction. So periplasmic, any protein that has a signal peptide. So we looked at proteins or predicted proteins with signal peptides, and we found the trend was again present there and even stronger. So in this case, we found the opposing trend, the expected opposing trend with lysine. Not so with histidine and arginine, but these are not typically associated with halophiles. So people are usually looking at lysine rather than histidine and arginine, as possibly because histidine is not present, as you can see here, at very high ratios. So changing that doesn't gain a lot. Its charge switches around physiological pHs as well. With arginine, it's quite useful in protein translocations, and its codons are quite GC rich, and GC is something you tend to see increase in halophiles, so it might be counterproductive to actually get rid of arginine at higher salinities. So So we're going to ignore those for the moment and focus on glutamate, aspartate, and lysine. Now, with the sold-out approach, you'd expect, as we see here, a strong trend or a stronger trend in the secreted fraction and a weaker trend in the cytoplasmic fraction. And so to look at the cytoplasmic fraction, we looked at ribosomes, and you can indeed see here that the trends kind of fall apart when you look at that fraction. And with glutamate, you can still see a little bit of a trend. So there is something still happening in terms of the cytoplasm. So there's still pressure to have an acidic proteome inside the cell, but it's substantially less. Okay, so next we looked at, this is data based on community averages. So the average of genomes across these different salinities. So we also wanted to look at this on a per organism basis. So these plots here, there's quite a lot to, there's quite a lot going on, but here we have each of these individual dots here is a genome of an organism that is most abundant in a particular habitat. And so we have all proteins secreted and ribosome. Ooh, and I'm just going to highlight the secreted fraction because we already know that the trend is strongest there. So in the middle, we have secreted aspartate, glutamate encoding on the bottom, freshwater, brackish, and marine taxa in sediment and water. And you can see there's quite a spread amongst all the taxa in terms of their amino acid encoding, but that it trends up in terms of the average across these habitats and with salinity. I'm just going to zoom in on aspartate here because it's particularly interesting. All these little purple dots here, those are actinomycetota. And you can see that on average, or above, they tend to be higher in their aspartate encoding. So that's a lineage-specific bias that's unrelated to salinity. But in spite of that lineage-specific bias, you can still see that the actinomycetota in freshwater have lower aspartate encoding ratios than those in the marine environment. So that we still see this adaptation related to salinity. Cool. We see those trends are evident within members of other dominant phyla as well. So up top here, we again have the actinomycetota, but down here we have our Bacteroidota and our Proteobacteria. So these are other major phyla that we found in the estuary. And you can see that in spite of lineage, that for each of these phyla, we have an increase in encoding with salinity in both environments, particularly for our acidic amino acids. Okay, so one of the most abundant organisms in the brackish environment was actually an actinomycetota called Aquiluna. Now, this is a little bit weird because Aquiluna is from the lunar cluster, and the lunar cluster is a group of organisms that are much better known as freshwater bacteria. So they're typically found in freshwater. So it's a little bit weird that we've got a saline adapted, or what appears to be a saline adapted lunar cluster member, this Aquiluna here. So this is an abundance plot with salinity, and you can see Aquiluna is really abundant, particularly in that brackish zone, but also in the marine reaches of the estuary. And here we have an opposing trend with a sister lineage, and this is Rhodoluna, so another member of the lunar cluster that is displaying what you would typically expect to see, so a freshwater distribution. So this is interesting. As I mentioned, these switches between freshwater and saline are thought to be quite rare, but this seems to be an example of where a lunar cluster member has jumped that divide. And as you can see here in this plot of glutamate encoding in our brackish environment, here's our aqualuna with the star around it, has much higher encoding than these little white dots, which are the rhodoluna group. So it has changed its amino acid encoding profile. Okay, so we wanted to explore this organism more in terms of its salt adaptation traits. So we looked at other traits associated with salt adaptation, so osmoregulation, for example. So this little heat map shows transcripts. So the red indicates increasing transcript abundance, and you can see here increases in transcripts associated with the salt-out approach or osmoregulation. So sodium transporter, so pumping salt out of the cell, pumping osmoprotectants into the cell, and an osmosensor MTR. Curiously, we also have one of the most highly expressed genes by aqualuna were elongation factor genes associated with, translation, protein synthesis. But these are also have been found in Arabidopsis to be associated with salt stress. So we think this is more of a salt stress signal. Okay. So what about amino acid encoding? We looked at this with the transcripts as well, and interestingly we actually found a really strong difference in the transcriptional modulation of codons encoded with salinity by aqualuna. So you can see at low salinities in fresh water it was encoding at much lower acidity, and in saline environments it's coding much higher acidity or more of these acidic amino acids in its transcriptome. So it kind of looks like aqualuna can actually modulate its transcriptional profile to help tolerate or help confer robustness to its proteome. Interestingly, we also found that with the basic amino acids encoded by the transcriptome, all three. So I mentioned that arginine and histidine, you don't tend to see a strong signal with those, but in this case, we found all three were much lower in the saline environment. So a really, really strong trend there, which I'll explain a bit further in a bit. This plot down here just shows aspartate synthesis with increasing salinity and 3 different pathways for glutamate synthesis as well. So we have quite a lot of activity in terms of glutamate synthesis in these saline environments for aqualuna. When we compared to rhodoluna, our freshwater, our sister lineage, all of this kind of again falls apart. We don't have any trends in terms of transcripts and amino acid encoding. It's pretty apparent from this that rhodoluna expects to live, or what I think is happening anyway is that rhodoluna is expecting to be in a freshwater environment, and only a freshwater environment is probably pretty unhappy to find itself all of a sudden washed down into the brackish reaches of the estuary. Whereas aqualuna, which is a brackish bacterium, is used to is adapted to both the brackish and the marine environment, and sometimes finds itself in the freshwater environment and seems to tolerate that quite well. In terms of what's going on with aqualuna's basic amino acid encoding, you can see here with these plots, we have lysine, arginine, and histidine, and expression in fresh and saline conditions. The gray dots here are all transcripts, and the colored dots here are differentially expressed transcripts, we see the strongest signal. We tend to see the strongest signal both for basic and acidic amino acids when considering all transcripts. These arrows here indicate the genome averages, and you can see that the genome averages pretty much match the transcriptome profiles for saline water, which is kind of what you would expect given that they're saline adapted, and are very different, so very high when in comparison to the genome average in that freshwater environment. So something weird is happening there in the freshwater environment. So we can look at what that is. So this plot shows the functional breakdown of transcripts across the salinity gradient from freshwater to marine. And you can see in these freshwater samples here that we have very low diversity of our transcripts for Aquiluna, so primarily translation, and these blue bars here are primarily associated with protein translocation or amino acid transport. If we look at that in comparison to the genome, so here in these pie charts showing differentially expressed genes in freshwater for Aquiluna and in saline water, and this is the compositional profile of the genome in terms of functions. You can see that the differentially expressed genes in saline water pretty much match the genome. So that makes sense. Again, if this organism is brackish adapted, it's pretty much expressing what it has in its genome. In freshwater, of course, it doesn't look happy. This looks like a stress response. So we have primarily genes differentially expressed associated with translation. Most of these are ribosomal protein-associated genes that are being expressed. And the thing about ribosomal proteins is they tend to be basic amino acid rich, and they're basic amino acid rich because they have to bind mRNA, which is negatively charged. And so that's not just true for Aquiluna, but for other organisms as well. And so the fact that Aquiluna is highly expressing or preferentially expressing ribosomal genes in the freshwater environment is giving it that really basic profile in that freshwater zone, which is potentially helping it tolerate that low salt condition. Cool. So what are those ribosomal proteins doing there aside from being basic? So probably not doing a lot in terms of actual protein translation. So we have, in terms of gene expression, again, with these heat maps, there's not a lot happening in the freshwater environment in terms of tRNA biosynthesis. So again, probably not a lot of protein translation happening. Also, here we have our ribosomal proteins, so you can see some of these are preferentially expressed in the freshwater environment as opposed to the saline environment. But these ones here with arrows are associated with the inhibition of protein synthesis. And so what Aquiluna is probably doing in the freshwater environment, we can only speculate, of course, with this transcription, is focusing on stress management and potentially also poising cells for a return to favourable conditions. Okay. And with that, I'm going to leave salt behind and move on to thermophilic adaptations. I'm going to keep with the same sort of theme though. So we're going to talk about acidithiobacillus and the traits that it has for tolerating high temperature and how those differ from its mesophilic relatives. And so this is work undertaken by a PhD student of mine. She's finished now, Kitty Sriaporn, and we published in Microbiome. And so in this study, we sampled the Taupo volcanic zone 6 different geothermal areas, a whole lot of hot springs, different temperatures ranging from 18 to 93 degrees Celsius, pHs mostly acidic. The Taupo volcanic zone tends to have acidic hot springs from 2 to circumneutral. And we sampled subaqueous sediments and also as part of a separate study, these digitates into this siliceous sinter that's deposited around the rim as well. Now, acidify-bacillus are better known as mesophiles. There's, I think, one species that's the cyclophile. So that's this is the growth range, the growth temperature range of acidify-bacillus. That's where you get your 4 degrees C there. And there's one moderate thermophile, which was isolated from coal spoils, so not a hot spring environment, but primarily mesophile, so happier around, in the 30 degree to 40 degree temperature range, and from environments such as, again, coal spoils, metal mines, and acid mine drainage. And the aerobic autotrophs, in the Taupo volcanic zone, we found them at temperatures up to 40 degrees higher than this. And we found them in many different hot springs. So again, not an environment that they're typically knowing from, but what we can say is if you sample a hot spring in the Taupo volcanic zone, you're very likely to find acidify-bacillus. So we've looked at cosmopolitanism in Taupo volcanic zone, TVZ hot springs, and there are a very small number of lineages that you tend to find over and over and over again in these hot springs. Those are actually shown in this graph here, which shows prevalence of, this is amplicon sequence variants across samples, and abundance in the community, and these colored dots here are the variants that you tend to, bacterial and archaeal variants that you tend to find across the vast majority of samples, and they also tend to be very highly abundant in the communities as well. Two of those that I've highlighted here are acidify-bacillus. The others are Thermoplasmatales, Disulpharella, and Mesoacidotoga variants. And so from our work and also work of colleagues, so Matt Stott and Craig Carey, who studied water in hot spring water in the TVZ. They sampled 1,000 springs in their case, so more than us. They found acidify-bacillus in 70% of water. We found acidify-bacillus in 90% of sediments and sinter, and the abundance across those environments ranged from 7 to 10% on average. In terms of the composition of acidify-bacillus communities in these environments, or in particular in sinter and sediments, it's shown here in this plot. Everywhere you see just white, that's where we didn't find acidify-bacillus, so you don't see very many white spaces here, because we found acidify-bacillus in almost every sample. And there's not very much going on here in terms of color distribution, and that's because there are primarily 3 main variants that we found over and over again. So who are those? They represent, so these are genomes we reconstructed from these hot spring environments. And our variants are shown in pink and purple. The purple one is a low prevalence group, but we're showing it here anyway. Green and yellow. Using genomic average nucleotide identities to infer species thresholds, we identified 4 species, so 2 here, 1 here, grouped with this other acidify-bacillus. This is actually and previously uncharacterized acidify-bacillus from a Taiwanese hot spring. And this yellow group down here are actually part of a previously characterized species. So Acidothiorbacillus coldus. So Acidothiorbacillus coldus is actually the only other thermophilic, or moderately thermophilic Acidothiorbacillus, and this one was that one from those coal spoils. So not a hot spring, these guys, not hot spring Acidothiorbacillus, but still thermophilic. And so what we have here is a clade of thermophilic, or a group of thermophilic Acidothiorbacillus, and up here we have our mesophilic relatives. And for our TVZ Acidothiorbacillus, you can see here with the coldus and the green clade and this pink clade here, that this y-axis represents temperature and the x-axis represents pH, that we have very broad distributions across temperature and pH. Okay, this graph shows that again there, for each of those groups, the temperature range. And these little black lines here indicate the predicted temperature growth optimums. And you can see that's all around just over 50 degrees for all the TVZ species. And it's pretty much the same for A. coldus as well. All the others are lower, so they're typically under 40 or around 40, and have much lower known growth ranges. Now, what we infer from this is that our TVZ hot spring Acidothiorbacillus are probably happiest growing around, like 40 to 60 degrees Celsius. And when they find themselves at either end of this extreme, they're probably not so happy, but they are able to persist. And so we do find them in those environments. Okay, so what are their adaptations? And so to look at that, we took our 19 environmental genomes that we constructed from the Taupo volcanic zone, and we compared those to 34 Acidothiorbacillus genomes from the genome taxonomy database, including A. coldus and mesophilic representatives. And we looked at protein encoding, so for all these codons and the predicted protein sequences. Again, proline is considered to confer rigidity to proteins to help with thermostability. And then we also looked for other traits that are associated with thermophiles. So these traits here are associated with genome streamlining, so genome length, number of paralogs or proportion of the genome that's in paralogs, and GC content, and also optimum predicted growth temperature. So what did we find? Oh, we found this. So this graph here shows percentage of protein encoded by the genomes. So each of these dots is a genome. And this is shown against predicted optimal growth temperature for the organisms. And if I highlight all our thermophilic species, or our hot spring group, those are all encapsulated by the circle here. This black dot, which is pointed out by the arrow, is the Taiwanese hot spring uncultivated organism, or the genome from that. In yellow here, we have A. coldus from non-hot spring environments, and in solid colors, we have our hot spring Acidothiorbacillus from the TVZ. And you can see that on average, they have higher encoding of proline compared to the mesophilic Acidothiorbacillus. Okay, if you're wondering how we predicted these optimal growth temperatures, we used a program called GrowthPred, which predicts these temperatures based on these other amino acids, which are correlated with growth temperatures. Cool. We also use growth to predict minimum generation time. So this plot shows minimum generation time in hours up the y-axis, and this is just plotted against genome length. Now, thermophilic bacteria and archaea can tend to grow at higher rates. So if you're growing at a higher rate, you have a lower minimum generation time. And that's probably associated with higher enzymatic rates in cells. And so thermophiles tend to grow faster, even though life is challenging at high temperatures. And so if we point out our hot spring bacteria here, you can see that they tend to have slightly lower minimum generation times, or in other words, they tend to grow faster. So on average, around 5 hours minimum generation. Other acidophilobacillus or mesophilic acidophilobacillus are here and sitting at around 6 hours. Acologist from non-hot spring environments is a little bit weird at sitting sitting down here with very low minimum generation time, so grows predicted to grow very, very fast in comparison. All right, so what about genome streamlining? So genome streamlining is thought to be associated with high temperatures because of, aside from growing fast at high temperatures are challenging. And so cells tend to, the cell sizes tend to be small, and that's thought to be associated with reducing non-growth associated maintenance. So if a cell size is small, the genome must also be small. And so what we would expect to see in our hot spring acidophilobacillus and other thermophilic acidophilobacillus, which is acologist, is that they're going to have streamlined genomes, so smaller genomes. And that's indeed what we see here. So we've plotted our estimated genome size against optimal growth temperature. Our solid dots here again are our hot spring acidophilobacillus. And this is acologist out here with slightly bigger genome sizes. But down here with our TVZ acidophilobacillus, you can see that they have genome sizes of around 2 million base pairs. Other acidophilobacillus are right up here between 3 and 4 million base pairs. So that's quite a big difference. When you get to, this is kind of the normal range for bacteria. When you get down to around 2 million base pairs, you're definitely starting to think that an organism is streamlining and living quite a reduced lifestyle. And so it's definitely in that reduced lifestyle range. You also expect to see a smaller percentage of paralogs or duplicate genes, and that's what we tend to see as well. So in our TBZ hot spring bacteria, they encode fewer gene duplicates, or a smaller percentage of those in their genomes. Okay. What when you have genome streamlining, one of the traits that is associated with that is what tends to be GC content. GC content tends to go down when you have a smaller genome because there tends to be less repair of the cytosine deanimation, so cytosine being converted into uracil. So you end up with loss of GC and AT-rich genomes. At 2 million base pairs, you wouldn't expect it to be very, very low, but you wouldn't quite expect to see what we're seeing here, which is GC content being a bit higher in our, or higher actually. Our TVZ group 1 group has the highest recorded GC content for acidophilobacillus, sitting at 62.8%. On average, which was a little bit surprising for us because it's going against that trend. If we replot that against optimal growth temperature, we again get this same expected trend considering that relationship between genome size and growth temperature. And here you can again see that trend where our TVZ group or a hot spring group tend to have these higher GC contents. The trend's a little bit messy, but it's again not what we would expect. So that triple bond with GC, we would potentially what explains this is the triple bond in GC conferring thermostability at higher temperatures. Now, other studies have been inconclusive with respect to GC cont and higher temperatures. However, based on this, we think it's certainly worthwhile to consider when looking at thermophilic traits or traits in thermophilic organisms. And with that, I would like to summarize. For our work with salinity, we have used our environmental genomic data and also transcriptomics to show that brackish bacteria, those guys living at really low salt concentrations across diverse phyla, indeed use the salt out approach, and as part of that, they encode acidic secretomes. For our work in hot springs with thermophilic Acidithiobacillus species, we show that they encode proline-rich proteomes and maintain streamlined genomes as part of thermal adaptation. And I would like to thank all these very wonderful people who have helped with this work, both for our salinity work and our temperature work. Not everyone is, of course, named here. In particular, I'd like to thank Dini Dinendu-Nayaka, who is from NESI, who has provided a lot of computational support to our group. And of course, I would like to thank our funders. So Australian Research Council grant co-funded the temperature work, and a massive thanks to Genomics Aotearoa for funding both lots of this work.
Thank you very much, Kim. Yes, have a nice afternoon.
Thanks, Mark and Peter, and everyone. And I'm gonna go home now because it's 5 o'clock.